Esperando Jack Sparrow.
Sparrow ha que està en directe, Potser té delay.
S'ha d'acceptar o alguna cosa?
No, refresh.
Potser comença a les 12 en punt, no? Ja són les 12.
No, ja són les 12 ja. Ara refresh.
Què feu ara ja?
Ho va provar i funcionava, sí.
Què ho feu aquí?
No. Ara. Va amb delay.
Me estáis tapando. No para. Sí, por aquí delante. Allá, ¿no? Venga, va, pasa. No, el no pasa en el computador. Sí. Y la voz.
Con el frío nos vamos adaptando a todo.
Thank you, Anna, for introducing me, and thank you so much. Today I'm going to present my PhD thesis, Integrated Genomics with Adaptive and Quality Tracing Mechanisms Red Week, Genetic Insights from Analysis to Model Development. So today the thesis will be general introduction, methods, and 4 chapters and conclusions. So wheat is one of the most widely grown crop on Earth, cultivated on more than 220 million hectares across all the continents. This map illustrates the global distribution of wheat production from 1994 to 2023. Major producers are such from China, India, and United States, and part of Europe are represented in dark shades. And wheat is not only the widely grown, it is also a fundamental staple food. It provides roughly 40% of world's population with a major share of daily diet. This consumption map shows the Asia and China particularly accounting for highest wheat intake, and overall wheat contributes about 20% of global daily calories protein intakes, and making it as the most nutritional significant crop. And looking ahead for the global wheat demand, expecting to rise sharply with world population projected across over 9 billion by 2050. Wheat production mostly increased by approximately 60%. However, the production faces major challenges like climate change, disease outbreaks, and lack of genetic diversity. All threats our ability to meet the food needs. In the Mediterranean basins, climate change represents a serious threat to wheat production. The models predict the rise of average temperature from 2 to 6 degrees Celsius during the June to August, combining with a 20 to 30% of reduction in rainfall from April to September. Since most wheat is grown under rain-fed conditions, this system is highly vulnerable. This is why Mediterranean is considered as a climate change hotspot. It provides an ideal natural laboratory to study adaptation and resilience in the wheat. And drought and heat stress are becoming more frequent and severe. They particularly impact the crop during flowering and fruiting durations. The most sensitive stage of the yield is the rainfall duration. The result is a reduction in both grain number and grain weight. So wheat phenology, the timing of flowering, heading and maturity is a crucial adaptive mechanism controlled by VRM, PPD and PPS genes. These genes determine how wheat responds to vernalization, day length and earliness. The early heading and anthesis allows plants to escape the terminal drought and the heat stress while maintaining the longer rain-feeding duration phase, securing the yield. In an environment like Mediterranean, water becomes the scarcity after flowering, therefore the plants rely more on stored carbohydrates from the pre-anthesis phase. For breeding, that means selection of early flowering genotypes with extending rain feeding, combines with agronomic practice such as optimizing soil pH and irrigations. So the Green Revolution boosted wheat yield with semi-dwarf, high input varieties, but relied more on few allied parental lines. This narrowed the wheat genetic diversity as a result. The modern cultivar have much lower allelic richness and nucleotide diversity than the traditional land races, limiting their ability to adapt heat, drought, emerging diseases, and the quality. So Mediterranean land races are reservoir of adaptive genetic diversity in wheat. They evolved under farmer selection in drought-prone, low input environment and gaining stronger tolerance to heat, drought, and nutrient limited soil. Importantly, they also have the unique alleles like glutenin alleles associated for superior drought properties, and therefore the valuable source of integration into modern cultivars are land races. So the breeding focusing for climate adaptation, quality, and yield improvement in wheat is necessary to study these land races. One of the main challenges in breeding programs is improving the genotyping for further yield enhance. So in early 1990s, when the RFFP markers were first used in wheat, they enabled the first genetic map and diversity studies, although the methods were slow and laborious, so often REBD markers followed by SSRA, black library cycle. So the highest genetic linkage map based on PCR markers were developed in 2000s, and establishment of International Wheat Genome Sequencing Consortium was done in 2005, and the first consequences map and the GWAS was performed in wheat in the same period. In 2010, the decade, the dark SEC array technology has been developed for wheat, and the shotgun golden gate SNP set was done, and the first genomic selection study was also performed, and 9PK, 9K SNP chipset was also being developed. In 2015, the reference genomes of wheat and wheat related species and cast markers were also being performed, and the present the wheat expression database, transcription atlas, and the wheat diversity panels, land races, and wild relatives were being developed according to wheat UG, and more than 10 wheat genome projects were being studied for capturing structural and allelic diversity. And pangenome graph explores variant across the 16 cultivars. So using this, due to this, the market development, the traditional wheat breeding relied on phenotypic selection. It can be advanced to genomics and whole genome marker predictions, where the breeding values early on. This led us to capturing small effects genes based on complex trait. It generally shortens the breeding cycle, so that we can move from traditional breeding to genomic selections. Instead of going phenotypic selection based, we can do the early selection on faster breeding cycles. So the key genomic selection success factors depends on marker density, training population size, and the diversity of the population, and trait heritabilities, and cross-validation for model accuracies. And the pipeline for the genomic selection is the training population and the breeding population. The training populations are being split into training set and testing set, where the training set is being trained by genotypic data and the phenotypic data, and estimate the marker effects throughout for the whole genome, and calculate the breeding values for test sets, and then do the cross-validation for the observed and predicted values, and using selecting the best models from the machine learning models or traditional models, we do predict the breeding values for the whole breeding population. The goal of my PhD was to strengthen Mediterranean adaptive breeding by combining genomics, genomics, and statistical breeding approaches. And the chapter 1 focused for identifying adaptive genomic regions in Mediterranean land races. linked to drought escape mechanism, environmental response using AHEN and environmental GWAS, whereas the chapter 2 is to dissect the genetic architecture of quality traits and evaluate genomic predictions for early selection of a complex traits in the same land races. And the chapter 3 focusing on characterization of high molecular weight glutenins, alleles and blend hardness variations, and assess the contribution of end-use quality in the same land races. And the chapter 4 is to evaluate the genomic selection models in interbreeding programs to identify robust prediction strategies of yield under variable environments. And in this study, I worked with two main wheat populations. First, the Mediterranean landscape land races used for first to third chapters. We use 170 land races from 24 Mediterranean basin countries, consist of three subpopulations, West, North and Eastern Mediterranean, and 26 were admixtures. The northern land races were longer growth cycle, whereas the southern were for early flowering, and it is a strategy for terminal drought escape. Whereas the chapter 4, it consists of 179 modern red wheat land races, red wheat genotypes, developed from multi-parental lines, and these parental lines were included also in the training populations. And the field trials were conducted in Geda, rain-fed environments for the land races, whereas for the genomic selection modern breeding lines were evaluated in two environments and with rain-fed and irrigated trials, and it was designed with randomized complete block designs for 2 replicates and 2 years. And the populations were done for genotyping by 15K luminosity per day in the land races and 25K luminosity per day for the breeding population. And the quality filtering were performed for both data sets. And the genomic selection model, we use the traditional RR glove, Bayesian models and RKHS models as a baselines, and then we compare with the machine learning models, random forest, upper vector machine, and XGBoost. And the cross validations were done within and across environments. The environments were water stressed and annotated as WS, and well water as WW, and we also combined both environments and also data predictions. For the GWAS, we used MLM with PCA and link shift matrix, and the threshold to select the market rate associations were 3, and the analysis was performed in facet. And the candidate genes were identified around the significant SNPs using the IWGSC reference sequence, and also the wheat expression analysis to see the expressions of the genes which we found. So the chapter 1 is published in International Journal of Molecular Science. And to understand how Mediterranean land races adapt to the native environments, we combined 15 years climatic data set from 23 countries of origin. We extracted the key environmental variables such as temperature, rainfall, evapotranspiration, solar radiation, and humidity. Instead of using annual averages, we aligned the climatic data set by 2 biological relevant crop windows from sowing to anthesis, which captures the vegetative phase, and from anthesis to maturity, which represents the reproductive grain filling phase. And we use 3 to 7 stations located in the main wheat growing areas of each countries. ANOVA showed very strong year environmental and genetic structure effects on the phenological traits. We see clear differentiation across seasons. 2016 was the shorter total cycle, but longest days to booting to anthesis and grain filling duration. And 2017 was the fastest development and shortest booting to anthesis and grain filling duration. And 2018 was almost complete longest crop cycle overall. Whereas we also identified the clear separation between north and southern land races by the climatic adaptations. And northern land races were for longer cycle and longer time to anthesis, but shorter grain filling duration. Whereas the southern Mediterranean land races were shorter cycle, and it is a classical strategy for drought escape. And we also identified the similar pattern by also the genetic subpopulation. SP2 subpopulation 2 were from northern Mediterranean land races, longest cycle and shortest grain filling duration, whereas SP1 consists of west and east, were also similarly as we observed with the climatic separations. The interactions were only significant for maturity and length in duration, for year and zones and year and subpopulations. This clustering analysis based on phenological trait clearly separates the land races with three climatic driven groups for the phenological traits and distributing emphasis and grain filling durations. So here group A consists of southwest Mediterranean land races, half early flowering. The lower values are for green and the higher values are for with the red, and the intermediate with white. And group B consists of northern Mediterranean land races, half longer cycles overall, and group C consists of southeast Mediterranean with intermediate cycles. Therefore, the longer cycles in cool, wet, north and shorter in high, dry were observed, and significant difference between the north and south, but not within the north and south was observed. And group B consists of northern Mediterranean land races, have longer vegetative phase and shorter grain filling durations. For the GWAS for phenology, environmental and population differentiation, we identified 306 marker trait associations detected across 17 chromosomes for phenological traits, and most associations were for heading, and 75 marker trait associations, and highest signaling was observed in 7A and 1B, with 60% at 208 centimorgan position, and with 1B having 42 marker trait associations. And for environmental GWAS, we identified 258 marker trait associations, and for sowing analysis we detected 63, and analysis to maturity 195. And the highest signaling was on 1D for temperature minimum across 19 centimorgans. And for alien GWAS consists of 10,458 SNPs. The top 5 alien values were explaining 22% of variation, which has a complex structure. 87 marker trait associations across 16 chromosomes were identified, and stronger signaling on 7A. So we also performed the hotspot discoveries for 567 initial positions. We had 158 peaks, these peaks, in the outer layer of these marker controls. And higher confidence filtering, we detected 69 peaks with 46 QTL hotspots. And the final set of more robust statistical methods, we detected 33 hotspots and 12 hotspots on A genome, 18 on B, and 3 on D. The majority of the hotspots were in A and B genome, considered as the B diversity triangle, which has less density of the SNPs on D genome and also higher linkage distribution. And these hotspots were also lying on this traditional genes. For example, 2A and 5A.3 were significant for phenological traits, and they were in photoperiodism and vernalization genes. In 1B and 1D, we detected on cytokine and flowering genes, and 7.8.5 we detected for grain color and carotenoids. And in 1B, significant for climate were in secaleae, so the transportation of rye into the wheat. And for the candidate genes, among these 33 stable QTLs, we identified more than 1,000 candidate genes, consist of 371 gene families approximately, and 42 genes among them were having differentially expressed genes among widely expressed experiments and the controls. And we prioritize these genes using the gene functions and relevance with the genes. For example, cell signaling, protein folding to be responsible for heat tolerance, and ABA regulations from controls. So the, among these 9, we selected 9 candidate genes stronger, and most of them are in 1B and 5B. And these are differentially expressed between non-stress and stress conditions for drought. And the roots, these 3 genes were highly expressed in roots in the drought, and they are responsible for ABA signaling and drought precipitation. Whereas for the leaves, they are for heat-induced protein stability and ROS defense, and in spikes, protein grain formation under terminal stress. So we also We use syntheny to determine whether the structural hotspot discovered in wheat occupies also in conservative genome blocks in other cereals. So we detected stronger conservations with bradypodium, rice conforming ancient stress response regions, and maize shows less syntheny due to greater evolution divergence, and these shared blocks likely consist core stress regulating genes, which increases confidence. And these loci are biological importance, not random statistical signalings. They strengthen our argument that these land races hold useful evolutionarily conserved values for drought and heat tolerance. So among 24 hotspots were being syntheny counterpart, and 97 markers in 79 syntheny regions, and 20 were orthologous QTLs, and 41 orthologous candidate genes, and one gene was common in all the genomes, and that was responsible for cell wall biosynthesis and otherwise mediated pathogenic defenses and osmotic stress responses. The chapter 2 was published in Frontiers in Plant Science, and this was performed with the collaboration with Dr. Carlos Guzman and CIMI to screen the landraces for quality traits. And the quality traits are largely genetically determined and group prospective for selection. We evaluated the grain quality, the ecological and agronomical traits among these 2 years, and the stronger genetic control for most of the quality traits were observed, especially for gluten strength, flow sedimentation, and flow rate, approximately 82% for gluten strength, and lower genotypic variance for test weight, grain hardness, and grain weight. Whereas the heritabilities were ranging from 0.6 to 0.9 for mostly quality traits, and highest for gluten strength 0.9, flow sedimentation 0.85, followed by flow yield of 0.81, and the lower for test weight, grain yield, 1000 kernel weight, and flow grain elevations. So there is G by E interaction for quality traits, the stronger G by E for effective for protein traits, whereas the minimal G by E was observed for mixing time, peak, and tendency by extensibilities. This plot shows the widely quality diversity preserved in Mediterranean landraces, mainly high protein landraces, tends to have higher P by L ratio and lower gluten strength. Although, but there was also important exception, for example, exception T371 having lower P by L and medium gluten strength, and also the case of group B, which have less than 11% of grain proteins. And also we also identified another landrace with having a higher gluten strength with almost 451 and higher P by L, and having a group 2 with more less than 12% of grain protein. So therefore, the conclusion of this phenotypic diversity of the landraces is we can introduce the alleles for the modern varieties, but we cannot move forward with the same landraces as a variety in the exception model. Therefore, the correlation matrix highlights the main trade-off shaping the grain quality in landraces. Protein and fluorine are expected perfectly correlated, but protein shows moderate negative correlationship with the E as we expected. And we detected the gluten relative traits having a higher correlation, especially for P by L, mixing time, and group volume. And the protein traits were negatively correlated with the gluten traits. And the grain hardness aligns with gluten quality, confirming the kernel hardness is also linked to the quality traits. And selection is important for based on balanced profile, high protein extensibility, and medium P by L, whereas the 1000 kernel weight it was negatively significant for the grain quality traits. Therefore, we have to be careful in selection of higher grain size may also weakens the gluten in these landraces, and integrating the quality phenotyping and genomic information to break the trade-off within the landraces at point. So loop volume is a critical target in wheat quality breeding, but it is extensive, labor intensive to measure, especially in the large population in the early generations. So the goal is to predict loop volume using simpler routine traits to enable large-scale screenings. So we developed 5 different equations. The equation predictions range from 0.74 to 0.90. Depending on the simpler traits, we were able to predict 0.74 of the loop volume, whereas we replaced the grain hardness with mixing time, we were able to predict 0.85. It's a practical model, we believe. because the brain protein and for segmentation is easy to measure and the mixing time is a little bit complex. Whereas we replace the mixing time with PVL and we have 0.84, which is almost similar. And the best model goes with 0.90 using ecological properties and flow segmentation. And we replace the mixing time with flow segmentation, but we were not able to predict higher than the previous models. So we detected 165 reliable associations across the genomes for most of the variation explained in A and E, as we were also observing in the chapter 1. And the traits explained from 7 to 31 of phenotypic variants, and A genome have 71 marker trait associations, and B genome have 78, and D genome have 16 marker trait associations, and 25 marker trait associations were significant for more than one trait. So it could be a tightly linkage or a pleiotropic effects, and mostly marker trait associations were observed for Dow strand of 20 marker trait associations with an allelic effect of 60 to 68, and only 3 marker trait associations for density by extensibility and test weight. And the shared protein was significant for 9 SNPs, were also significant for flow protein and the grain protein. And the yield, we detected only 14 marker trait associations, and 2 of them were mapped with Dow traits and quality traits. So the multi-trait marker trait associations and hotspots on 4A and 3B indicates the genomic regions, and 53 critical hotspots were identified, especially the hotspots in 4A consist of 50 marker trait associations, were significant for quality traits, gluten strand mixing time, peak and root volume, whereas the major hotspots in 3B with 13 marker trait associations for protein traits. And the yield-related hotspots were identified in 2B, 4B, and 5B. And a few hotspots explain multiple quality and agronomical traits are a key breeding targets. Within the window of 500 kb, we identified 807 genes across 52 critical hotspots, and 256 were differentially expressed and also expressed especially in the grains, and 29 were upregulated and 102 were downregulated during the grain development from making stage, soft dough stage, hard dough stage, and dough stage, and ripening stage. Among them, 10 grains were specifically expressed in the grains, and then we dive more deeper with the starchy endosperm and organelle layer as the key tissue for protein and starch developments. The results of agronomical traits, the evaluation of genomic selection within the land races, we tried the higher predictabilities for quality traits were observed, so we were able to predict higher quality traits compared to the agronomical traits. So the P by L having 0.29, and gluten strand was 0.64, and grain filling duration of 0.65, and flowering for 0.3, and the grain yield was the least of 0.52, 1000 weight of 0.42. Chapter 3 is published in Journal of Cereal Science. This was a study in University of Polytechnical in Madrid, and which was supervised with Dr. Patricia Giraldo during my stay. And we characterized the high molecular weight gluten subunits across the Mediterranean land races. As expected, we identified GLUT1 showing the greater diversity, and we identified totally 69 high molecular weight gluten combinations across 168 land races. Highest variability was as expected, we observed the GLUT1 loci. So the highest was for GLUT1, and then GLUT1, and then GLUT1 in the diversity, which was also observed in multiple global states in the land races. So this was a representative gel within the land races. So we can see higher diversity within the land races. And we observed a rare null allele, especially in GLUT1. It should consider as like this 2, but we observed only 1, and we also observed a rare null allele here in GLUT1 loci. And we also observed a rare combination of 32-33. There were observed only 2 Italian land races, and we observed a new, which was not even documented before in any publications, which have less mobilization than 7, higher mobilization than 6. So this null allele and rare alleles indicates the rich gluten gene diversity preserved in land races. So the geographical distribution among the blue ribbon loci, 2 star was widely spread among 22 out of 24 countries and dominating mostly in the North Africa and Turkey. And subunit 1 was 17 out of 24 countries, especially in the Northern Europe. Null is richness in North Africa and the Middle East. And the red 2 dots is in Spain, Syria, and Iraq. So the country-specific occurrence was also observed in only in these 3 countries. Whereas in blue 1, 28 plus 25 is present across the multiple countries. We couldn't find a clear pattern of blue 1 because of the higher polymorphisms. And 17 plus 18 and 30 plus 16 was only observed in Bulgaria and Spain, Portugal, and whereas in blue 1, 2 plus 12 was the dominant one and observed across the Mediterranean regions. And subunit 2 was only observed in Jordan, which have a null allele in blue Y. And subunit 10 was only observed in Turkey, and 2 plus 12 star subunit was found in Serbia. Whereas blue 1 is the main determination of bread making quality, so we wanted to see an ANOVA for checking the glucose significantly affecting both sedimentation and gluten strength. So glutamine loci was significant for both of the traits, and consistently with the known effects of 5 plus 10 having stronger gluten and 2 plus 12 having weaker gluten. And in glutamine 1 significantly influenced for gluten strength but not for flour sedimentations, and whereas in glutamine 1 was not significant for both. The phenotypic variation in the landraces, we just was to see few representative landraces from our countries. So widely flow segmentation and gluten strength variability having a stronger quality diversity. The landraces which have null allele like GLUE1 and GLUE1 having lower flow segmentation were typically from the regions where the flatbread were being predominantly used. Whereas from Romania and Bosnia, these 2 landraces having higher gluten strength and gluten volume, they are the regions from more stronger baking regions. Another landrace having an intermediate gluten strength, they were from Algeria and whereas the variations in the purulins, we identified 7 purulin genotyping across all the landraces, and we identified a landrace which having a nucleotide change from tryptophan to deletion of 68 SNP, and also we identified a deletion at the position of 73 and a stop codon at the end of the value. So we annotated them. And the highest frequency of this purulin genotyping was observed the soft wheat with having a frequency of 88 across all the Mediterranean regions, and the purulin A and pin B D1B was only observed in Italy and Algeria, and whereas the Iraq have only the allele with having pin D1B and pin D1A, whereas in Israel and Jordan having the nucleotide change allele, whereas the deletion allele was from Algeria and Romania. Therefore, we also screened the landraces with wheat bread making gene, which is getting attention in the recent days, which is responsible for loop volume. So we wanted to see if the landraces also carry these alleles, so it can also be a source of information into the modern varieties. So 12 of the landraces among these 178 landraces were positive for this allele, and among them 10 were having a higher loop volume, more than 600 ml. And bread making performance were variably from soft gluten strength. And we also identified this null allele in GLUE1 also having a higher loop volume. So we believe that the contribution of wheat bread making gene, allele, can be also an influence for higher loop volume, although we have a null allele in GLUE1 size. And also in another landrace which was having null in GLUE1 and null in GLUE1, also having a higher loop volume of 612. Whereas one notable exception is having a highest among all these 12, were having also a high gluten strength and also a high loop volume. And this landrace was from France. Another landrace from Spain was also excellent for loop volume and higher P by ml, and although we have a less gluten strength compared to the landrace from France. And the chapter 4. This was accelerated by methods recently, 2 weeks ago. This study used a training population of 179 modern bread wheat lines, evaluated for 2 years under both rainfed and irrigation conditions, and the phenotypic data includes yield and yield related parameters. And these were also evaluated rainfed and irrigations with 25 kg chips, and the quality filtering with imputation of missing genotype, missing information of 30%, and minor frequency of 5% will be avoided. And we want to compare the traditional RO biasing and RKHS models combined with machine learning models. And the cross-validation schemes, we use 2 cross-validation schemes, K-fold cross-validation, which have 80% as a testing and 20% as a training population for 150 iterations. And there are other cross-validations have one genotype is being predicted with the rest of the genotypes, till all the genotypes are being predicted. And the different cross-validations were performed with water stress and combining water regimes, and also we were interested to train on water and test on water stress and vice versa, and also train the models with complete water regimes and testing on individual environments. And the prediction accuracies were completed with Pearson correlations between observed and the predicted values across all the iterations, and this analysis was performed using RStudio. And the we observed lower activity for gradient water stress, which have a higher environmental influence, and then followed by well water, and then when we combine data set, we were able to avoid the environmental influences. Whereas the highest variability was observed for 1000 gallon weight up to 0.887 in complete water regimes. And the rain protein also have a stable and higher under well water conditions of 0.85. Whereas the correlation between the traits within well water and water stress, we observed a negative correlation between grain protein and grain yield, and a positive correlation between number of spikes per meter square with grain yield, and number of grains per spike was negatively correlated with 1000 gallon weight, and the same magnitude and tendency was also observed in water stress conditions. Whereas the correlations between water stress and well water, we observed a stable positive correlations except for grain yield, which have higher environmental influence of 0.35 correlation between water stress and well water conditions, and the same observed also for number of grains per spike with 1000 gallon weight and negative between water stress and well water, also here. And then almost all traits were well in machine learning and RLM, but not in biasing models. They were not able to predict well, maybe they were overfitting. And in the machine learning models, the random forest was able to predict similarly to the traditional model in RLM for well water conditions. So the RLM for grain yield under water stress was able to predict 0.76, whereas in well water conditions 0.75. The random forest was able to predict 0.74 in water stress and 0.80 in well water conditions. When we combine the both trials, we were able to predict 0.64 for grain yield under combined water stress for RLM here. And for random forest, we were able to predict 0.71 for grain yield. So, using the Livono cross-validation, we also observed similar trend as the K-fold cross-validations, which as the biasing models were not able to predict as compared to the RLM, so we avoid the biasing models and we move forward with the RLM as a prediction model and compare with machine learning model. So we observe similar trend with random forest and RLM having a higher prediction accuracies compared with support vector machine as the least and intermediate with HTB, although a small deviation of random forest has been predicted higher than the RLMs. Whereas if you compare the two cross-validations for all the traits among different environments, we observe a small deviation of K-fold cross-validation and Livono cross-validation for grain yield to water stress and the water conditions. Whereas for the plant height also the same within the same two environments combined with complete water regimes, we are able to stabilize within two cross-validations. But a higher penalty here was observed for 1000 gallon weight in well water conditions from 0.69 to 0.41 of the deviations for XG boost models. Whereas the prediction transferability depends on the training environment. So when we train within the individual environment and testing on the opposite environment, we were able to predict lower. Whereas when we train the models with a diverse data set by combining the combined water regimes and predicting on the individual. We were able to predict higher, especially for grain protein. And with the diverse data set, the RW were the lowest ones, whereas in the machine learning, the XG boost was the highest one, comparatively within the machine learning models. Earlier, the random forest was better in the individual environment, but when we have the diverse data set, the XG boost was able to learn and predict well among the diverse data sets. With the permission of the tribunals, I would like to move to the conclusions. Thank you. The differentiations in phenology across climatic zone underscore the genetic basis of adaptations to drought, temperature stress. Key QTL hotspots were identified as major regulatory, influencing flowering time and growth cycle, duration to enhance drought escape strategies. Environmental GWAS identified genomic regions significantly linked to environmental variables, particularly during the grain filling period, which plays a crucial role in climatic adaptations. Air GWAS revealed key differentiations loci among land bases of population, demonstrating the impact of natural selection in shaping genetic diversity and resilience. The synthetic analysis confirms the conservation of regulatory elements across the cereals, suggesting shared adaptation mechanisms that can be leveraged for breeding climate resilient wheat cultivars. The prediction of low volume using multiple linear regression improves the accuracy as trait complexity increases. A good balance between accuracy and trait complexity allow efficient predictions of complex traits. The significant marker trait associations and critical hotspots were identified for key quality traits, providing promising targets for marker-assisted breeding and trait pyramiding to optimize wheat varieties for industrial and consumer needs. Genomic selection using traditional models demonstrate moderate to higher prediction accuracies for quality traits such as lower ecology, supporting its application in pre-breeding programs. Genetic variability in high molecular weight glutenins among land races was extensive, with group B1 showing the highest polymorphism. Gluten in subunit distribution varied geographically, reflecting selection pressure and adaptations. Novel alleles for purulin D1 have been identified in Mediterranean land races, potentially affecting kernel texture. Genomic selection was more efficient under irrigation due to improved heritabilities and prediction accuracies for key traits like grain yield and grain weight. Machine learning models showed higher adaptability in predicting grain yield and number of grains under variable conditions compared to the traditional model. The training on combined data sets, rainfed and irrigation, improved model robustness and transferability across the environments. I would like to take a moment to thank and acknowledge all the firstly, my supervisors, Dr. Jose Miguel Soriano and Dr. Marta Silva Lopez. I was the FBI Fellowship for my grant to perform this study. And also the Sustainable Extensive Field Crop Group, and also my scientific mentors and collaborators, and colleagues and friends, and my family. Without them, I would not be able to do this book. And thank you so much, and thank you for your time. Thank you for your time.
Also, I just to give a general overview of what we should take as a home message. But anyway, it's not really bad here. But anyway, I find it very, very nice. So yes, of course, I would like to congratulate you because of the hard work. I think it has been quite successful. I also would like to congratulate Jose and Marta, the supervisors. I think that you did an excellent work too. But the merit is basically yours, of course. And yes, I have some questions. I'm not an expert at this anymore on Syria, so there are going to be some general questions on the topic. Some of them may be obvious, so I apologize. Some others, well, I have some curiosity that I want to share with you. And the general one that I would like to know your opinion is regarding the first chapters in which you are using land races, which as you know better than me, they are not homogeneous material, genetically speaking. So they consist of a mixture of pure lines. And so there is variability within each land race. And if I understood well, you handle this variability differently across the chapters. For instance, there is the first chapter in which you say that you choose the dominant type in order to genotype the land race, like focusing on a dominant type, which I don't know if this dominant type is already a pure line or still is a mixture of things. In another chapter, you say that you missed what you do was to take 5 grains per land race in order to be genotyped. In this case, I don't know whether these 5 grains come from the same plant or from different plants, so trying to catch the variability within the land race. In summary, so how did you deal with this variability at the land race level in order to genotype and to get conclusion on this genotyping at the land race level?
Thank you, Walter, for your kind words and for the thesis and congratulating me. But as you said, yeah, I will go step by step.
Yes.
So I have a small slide for PhD and future directions, and yeah, the limitations and the future directions to validate the key genes, and the take-home message is to validate these genomic regions for phenology and identify the quality alleles different for the hematopoietic glutamines and the promalin genes, and the genomic selection plays book for the readings. And yeah. No, I just kept it aside because of the limitation of the time. Because I was on time. I really want to take one more.
No, it was okay. I mean, we are very well.
Yeah.
Yeah,
and also for the question, the structure of the thesis, we did the genomic selection in the land races in the chapter 2 for the agronomical traits and quality traits. So we want to see if the same will be translated also in the modern varieties, because we were having a breeding population from Irtha, which consists of multiple parents. So we were interested to see how the genomic selections are performing in the modern varieties by the same information or the same knowledge behind, within different water stress conditions and different models. So we were interested to go specifically for that. And regarding the variability within the land races, in the chapter 1, we genotyped the dominant plants within the plot, but for the replications of 3 replications per plot, we send the DNAs to the service, and we have selected the homogeneous alleles from them. And Conchita, the previous researcher from our group, she have